So you should have heard a little voice there that says this session is being recorded, which means this session's being recorded. It means also that at some point in the future, maybe in the next couple of weeks, I'll send a link round so you can watch this back if you want to re-watch it. I'll also send a link to all the other ones that we've done as well. So if you didn't catch any of them and you'd be interested in watching any of the ones that haven't happened or are going to happen, you can see those as well. Now, I can see the numbers are on my little counter are going up to say that we're getting lots of people logging in, which is great. But it normally takes a minute or two for that to happen in Zoom. So whilst we're doing that, purely for fun, just because we're really interested to know where you're joining from, could you let us know in the chat where in the world you're watching us from? I'm in southeast London. Yesterday was a little bit sunny. Today it's kind of overcast, but yeah, I'm in southeast London. Somebody just said they joined from Japan. Well, that's very interesting. It's very interesting. Japan and Malaysia. Newcastle, cold and wet there. Somebody else from Malaysia. So it's really interesting for us because this is the only session we're doing at 9 o'clock in the morning. We have other ones that are taking place in sort of at lunchtime or later in the day. But I've noticed that we've got people from Thailand, Malaysia, Japan, and Newcastle, and Nottingham. So this is a very interesting spread of people. Fascinating stuff. Thank you for letting us know. That's really, really interesting and helpful. And somebody from Colombia. Well, very warm welcome from Colombia. So for everyone who's joining us from around the world, it's great to have it. And somebody from China. Yes, you're happy to be here. Well, we're very happy to have you. Right, so I'm going to launch a couple of polls which are really just to gauge the composition of the audience so we know a little bit more about you. It's anonymous, but the first question is whether you have an offer to study with us. It's just because when we send the invitation out to this, we send out mainly to people who are inquiring about studying at biochemical engineering or at UCL. But for a lot of people, we also send it out to people who have an offer to study with us. And it's interesting to look and see that we have most people so far, according to the poll, don't have an offer. So we're assuming that you're prospective students. Several do, and some do, but not. But someone else at UCL. So thank you for joining us. It's great that you're interested in our subject. I'll end that poll now. Appreciate your time. Appreciate you filling that in. It's very helpful for us. And the next question is just to check on what level of study that you might be interested in. So this is more for us to know whether you'd be more interested in doing an undergraduate degree or whether you'd be more interested in doing a postgraduate degree. It really helps us to know whether you're maybe an A-level student or currently a degree student. And interestingly, in this session, it's roughly split between prospective MSc and prospective degree, which is particularly appropriate because Petra, of course, is the admissions tutor for the MSc Biochemical Engineering program. That's very helpful to know. Really good. Thank you for filling that in. So now that I've ended the poll and you've given me that information, I'm going to introduce myself. My name is Kim Morgan. I'm the Outreach and Communications Coordinator here at UCL Biochemical Engineering. And I'm going to shortly hand over to Dr. Petra Hanga, who is the admissions tutor for the MSc Biochemical Engineering. And Petra is going to talk to you about lab-grown foods. I will turn my camera off whilst that's going on, but I'll still be here in the background. I'll be typing away. If you want to ask any questions, please feel free to do so at any point, but please use the Q&A function. If you're on a mobile device, you may not have noticed, but you have a series of buttons at the bottom of your screen. You can choose how to watch us, what configuration you want us. You can use the chat function, which some of you have found already, but you can also use the Q&A function. The Q&A function is really important for questions because it means that we can answer them in order. We can maybe group some of them together, and if you put them in the chat, sometimes they just get lost, so we wouldn't get around to answering them. So you can ask them at any point, but we'll come to them at the end of Petra's talk. But for now, I'm going to hand over to Petra. Thank you.
Thank you so much, Kim. And welcome, everyone. I hope everyone's here can hear me all right. Thank you so much for joining me. As Kim said, my name is Petra Hanga. I am an academic at UCL Biochemical Engineering and also the program director and admissions tutor for the MSc in Biochemical Engineering. So I am absolutely delighted to talk to you about my research, which is actually in cultivated meat and is one of the lab-grown foods that I will be covering today. So these are my contact details here. If you want to reach out to me afterwards, if you have some questions that you would like to ask or just get in touch and say hello, feel free to contact me on either of the platforms. Okay, so lab-grown foods. I mean, this is a very hot topic. As I said, it is part of my own research that I do within the UCL Biochemical Engineering department. Some of you might be familiar with it, but hopefully today you will learn some new things. Perhaps you're already interested in the subject or you're already doing some work in this area. So the way I structured today's virtual taster lecture, I'm going to talk about the motivation for producing lab-grown foods, first of all, so you get an idea of why these are needed. Then I'm going to do a brief introduction to the new discipline of cellular agriculture, which covers lab-grown foods. And then I'm going to go through different examples of lab-grown foods, how they're made, what kind of characteristics do they have, what potential benefits, etc. And then I'll end there and I'll take your questions. So it is important to acknowledge that the world's population is continuously increasing. We are now around around 8 billion, so 7 billion in 2020. But this number is predicted to continuously increase to reach almost 10 billion by 2050. So the question becomes, how are we going to feed 10 billion people by 2050? Particularly given that in today's world, and actually these numbers are probably even more now with what is happening in the world with the two wars, but there are people already that are undernourished, people that already struggle to have access to foods. And then also we also have to consider the fact that the land that we have available for farming is very limited. And farming, when I talk about farming, I'm referring to both agriculture and animal farming. So we already have limitations in place that don't allow us to do more. And if we do try to exploit the land even further, it could cause even more catastrophic effects on the environment. So one way of tackling this need for foods, and especially the need for proteins, which are absolutely necessary for our diet, is intensive animal farming. And this type of approach has been taken all over the world. Maybe not so much in the UK, but it is quite prevalent in other geographic regions. However, intensive animal farming looks something like this, where you have thousands of animals crammed in very small spaces. So when that sort of situation happens, the risk for zoonotic diseases, it's very, very high. If one animal becomes sick, then it's very easy for that disease to spread. And that means in for these intensive animal farms, it means a loss of money if they lose all those animals. So to bypass that, that risk of zoonotic disease transmission, most often these animals are fed antibiotics to avoid that transmission risk. However, that obviously comes with a lot of issues, because those antibiotics will eventually end up being ingested by the consumers. And of course, when animals are overcrowded, there are ethical concerns about the animal welfare. Some farms will cut costs by reducing the quality of the feeds that those animals get, etc. So all of those things will have an overall negative impact. And of course, it's worth mentioning that in order to get the space, the land to host all these thousands of animals, that means that there is a significant use of natural resources, and not just land, but also that water. And I've put here some statistics to give you an idea. And of course, to obtain that land, we're talking about a lot of deforestation that has a detrimental effect on biodiversity, which tends to be lost. And animal farming has been found to produce the same amount of total greenhouse gas emissions as transport worldwide. So going back to that question, how are we going to feed 10 billion people by 2050? It is becoming very clear that business as usual is not an option, and we do need a long-term solution. But that long-term solution needs to be able to sustainably, efficiently, and very importantly, safely help produce foods. So what if we could produce foods that don't harm the animals, don't damage the environment, so they don't need that deforestation, and they don't impact the biodiversity. They don't require antibiotics. They need a lot less natural resources, and we're talking land and water, and produce less greenhouse gas emissions and are more sustainable. So this is how the new discipline of cellular agriculture became. And this term of cellular agriculture was first introduced in 2016 by Isha Dattar, who is one of the founders of a charity based in the US called New Harvest. And essentially what it means is the production of animal-sourced foods using biotechnology and cell culture techniques. So rather than harvesting from the animals, we're using established biotechnology techniques to produce those foods. And some examples include anything that comes from an animal. So first of all, we're talking about meat, which is the most obvious, but then we're also sourcing eggs, we're also sourcing milk from animals, and also materials like silk come from worms, leather, it's essentially animal skin that we then process. And I've also put here vanilla, which even if it doesn't come from animals, because it is a plant, the way vanilla is sourced has a lot of ethical issues. And actually vanilla can be produced using biotechnology. Or at least the component that gives that specific taste of vanilla. So these are all examples of animal-derived or animal-sourced products that can be produced using cellular agriculture. So is this really a new concept? Well, you might be surprised to find out it's not, and I'm sure a lot of you know who this person is. So Sir Winston Churchill actually declared in 1931 that we shall escape the absurdity of growing a whole chicken in order to eat the breast or wing by growing these parts separately under a suitable medium. And this is basically what cellular agriculture is. It's about producing those animal-sourced products using biotechnology, using cell culture. So that means growing cells using a suitable medium and under a suitable environment. So exactly what Winston Churchill envisioned back in the 1930s. So in terms of lab-grown products, lab-grown foods, we'll focus specifically on foods in this lecture. They can be classified in different categories. Sometimes lab-grown foods are also referred to as alternative proteins. And the different categories are plant-based proteins, recombinant proteins. So plant-based proteins, I'm sure a lot of you are familiar, especially now in supermarkets. There is a very wide range of products that are available based on soy. or based on pea protein, etc. Recombinant proteins refer mostly to products like milk. Then non-animal cell culture, and those of you that are from the UK, I'm sure you're familiar with corn, which is a mycoprotein, but then products based on micro and macro algae also fall within this category. And then animal cell culture-based products, and this is where cultured meat falls within this category. And of course, some of these have already reached the market, so they're commercially available, like corn is commercially available. There are some other products based on mycoprotein that are commercially available. And then plant-based proteins, there's a variety of those type of products already available. However, recombinant proteins like milk, which I'll cover in this lecture a bit further along, they're still in the concept. There have been proof of concepts, and the same for cultivated meat. Right, so I'm going to take each of these categories and give you some examples so you get a bit of a better understanding. And the first category that I want to talk about are the cellular lab-grown foods. So cellular means that these products are basically, they're not based on cells, but they are based on organic molecules like proteins or fats that are produced by cells. So generally, these type of products would be made using the process of fermentation, combined with recombinantly engineered host cells. And those host cells can be a variety of cell types. They can be bacteria, they can be fungus, yeast, even algae that can be modified to using recombinant DNA technology to be able to produce certain proteins. So for example, let's say we want to produce milk, which comes from the cow. So that means the protein that we would be targeting would be a bovine protein. Right, so first example of this type of product is milk. And I'm not sure how familiar you are with the process of animal farming to produce, of dairy farms for milk. But essentially, those mother cows are kept in a continuous lactating state. So that means that 10 out of 12 months that mother cow will be pregnant, so they can produce the milk, which is collected regularly. However, these type of practices have reduced the natural life expectancy of dairy cows significantly from 20 years to about 5-6 years. And after that, these dairy cows are then sent towards slaughter for meat. So these type of practices, they are quite gruesome. The fetuses or the calves are then collected and essentially used, well, slaughtered and used in other applications. So there is an element of animal welfare concern there, and of course on the dairy cows as well. So through cellular agriculture, there is possible to produce milk without having to go through these gruesome practices. And at the end of the day, milk is a concoction of a protein, which is casein, and other components like fats and vitamins. So it could be formulated without having to be sourced from the dairy cow. So the practice is to use recombinant DNA technology and fermentation to produce that casein. And there are actually a few companies worldwide that are trying to attempt this. And they are making significant progress. So hopefully in the next couple of years, we will be able to source this type of milk. Another example in the same category is rennet. And rennet is a mixture of enzymes that are used to make cheese. So those enzymes that will turn milk into curds. And traditionally, rennet used to be extracted from the stomach of calves, which meant that those animals would have to undergo slaughter. But actually, believe it or not, today the majority of cheese making is performed using recombinant rennet. And this technology has been approved by the FDA since the 1990s, and it has been produced since then. And actually, rennet that is produced using recombinant DNA technology and biotechnology is of a higher purity, is a lot more consistent, and actually it's a lot cheaper than the rennet that is extracted from the stomach of calves. So I see this as a success story. If this was possible, I'm sure all of the other attempts, like producing casein for cultured milk, the terminology is still not established, but for milk that is now sourced from dairy cows, will be possible as well in the next couple of years. So other examples include egg white proteins, and this is a company called Clara Foods that it's attempting to do that. Collagen proteins, and also the same technology is also used to produce cheaper growth factors that can be used in media formulations for cultured milk, cultured meat production. And that is a company called Maltus Media, which is based in London. Another example, which I do want to cover very briefly as well, are hemp proteins that can be produced using recombinant DNA technology. And these are important because hemp is it's essentially the hemoglobin molecule that has different functions in vivo, but in the context of foods, it gives that red color of meat, and also it contributes to that taste, to that meatiness. So it is a very important component. And a lot of the plant-based burgers or plant-based products, they are trying to achieve that meatiness. So they have been trying to use components that can replace this heme, because obviously hemoglobin comes from blood, so it is an animal-derived product. And plants do not have hemoglobin, but they have a version of hemoglobin, which is called leghemoglobin. And this is quite an interesting compound because it is found in legumes, and particularly in the roots of legumes, in those nodules, as you can see in this image here. And these have a very, very important role. They essentially work in a symbiotic relationship with a soil bacteria. So the plant provides the oxygen that it transports through this leghemoglobin present in the roots. And it provides that to the bacteria, while the bacteria transforms the nitrogen to ammonia, which is something that the plant will be using. So there is this symbiotic relationship. So companies like Impossible Foods have used this leghemoglobin as a replacement for hemoglobin or myoglobin, which is present in muscle, to achieve that meatiness of their products. The other category of products that I want to cover are fermentation-grown foods. And these have been around for a very long time now. And I'm sure a lot of you are probably familiar with these. Tempeh is one such product. Kefir is also another product. And what's really interesting about these products is that they are based on microorganisms, on live microorganisms that essentially have a really critical role. So in tempeh, for example, there is a fungus from the species of Rhizopus that ferments soybeans, and that's how tempeh is produced. Kefir grains, on the other hand, is another interesting example of a symbiosis, and quite an unusual symbiosis between different microorganisms. So between different species of yeast, but then also different species of bacteria. So tempeh, just a bit more about this. It can be made using any legume, bean, or grain. Generally, wheat, rice can be used. And the microorganism is the important one. So as I said, it's a fungus from the Rhizopus. And there are certain species that are used. And this fermentation is based on a solid substrate, which is the legume, the beans, or the grains, which provide the nutrients to the fungus, the microorganism. And tempeh generally is produced using solid state fermentation. This is quite a cheap process that takes place in trays. And the whole idea is that these trays are placed in a controlled environment that is it provides all the conditions necessary for the microorganism to grow and ferment that solid substrate, the beans or the legumes or whatever is being used. And it's called a solid state because there is very, very little free water available. So everything is done in a solid state. Another product of fermentation, but this time is based on biomass fermentation. So it basically leverages the growth of a specific microorganism to produce large quantities of proteins, is corn, which is a microprotein. And as I said, if you live in the UK and actually even in the US, you might be familiar with corn, maybe you even tried it. You know that over the years it has become better and better. And these are some images on how it looks like when it is produced. So microproteins, which are basically based on mushrooms, have been used as food for decades. It's quite an interesting product because it can be manipulated to mimic some of the organoleptic properties of animal muscle. And you can see here some scanning electron microscope images of different type products, including poultry and corn. And you can see how these are aligned in some respects similarly to poultry, perhaps not as tight, but they do provide the mouthfeel of meat and that texture of meat, which is quite desirable when we talk about products that are trying to mimic meat. And the last category of lab-grown foods that I want to talk about are the cellular lab-grown foods. And this is basically cultivated meat. So these are products that are made of actual whole and live cells. And generally, they are produced using tissue engineering techniques. Even though that trend or the manufacturing of these type of products, because they are so novel, is now maturing to become something that is at the interface between tissue engineering and food processing. So cultivated meat, you might have encountered it under other names like cell-based meat, cultured meat, clean meat, lab-grown meat, etc. But they basically mean the exact same thing. It is genuine animal meat that is produced without having to slaughter the animal. In a controlled way, so in a controlled environment, in a bioreactor, using cells that have been harvested from the animal. And cultivated meat, because it is genuine animal meat, it's meant to have the same composition as animal meat, the same taste and nutritional profile. And actually, because it is produced in a controlled environment or in a controlled manner, it can be tailored for improved nutrition. Excuse me. So that means that it could potentially be supplemented with good fats, omega-3 oils that are not naturally found in meat, or even other supplementations that can enhance that nutrition. And there have been some world first proof of concept. In 2013, Professor Mark Post, the founder of the startup Moza Meat, that is based in Maastricht in Netherlands, has showcased the first proof of concept cultivated beef burger. And then since then, a lot of other proof of concepts have followed, including Memphis Meats meatball, Finless Foods fish patty, Shiok Meats shrimp dumpling, Higher Steak, which now is called Uncommon, bacon, cultivated bacon. And of course, there have been many, many other more. But what I really wanted to show through the slide is the variety of products that can be produced within the cultivated meat umbrella. You can have non-structured meat, like anything, so like minced meat, anything like a patty, like a meatball, burger, etc. Or you can have structured meat, like bacon, which is a lot more structured, or fillets, or steaks, etc. And they can be beef, they can be fish, shrimp, anything that essentially replicates meat. We have in the same meat portfolio. And actually, there have been some really big milestones in the cultivated meat industry, with several regulatory approvals for commercialization. So in US, there are 2 products that have been approved, both cultivated chicken from Upside Foods and Good Meat. In Singapore, there are now 2 products as well, a cultivated chicken from Good Meat, and also cultivated quail from Val. In Israel, Aleph Farms have have essentially got the green light for the first step in the approval in Israel. And so far, they are the only ones that have done beef, but they have done structured beef through their 3D printed steaks. And of course, there are other parts of the world. UK as well has received several applications for regulatory approval. So in the next, perhaps even towards the end of this year or next year, I'm sure we will see many more cultivated meat products that will receive regulatory approval and will start being commercialized. Right, so how can cultivated meat be produced? The production is very similar to, as I said, it has been adapted from tissue engineering. So that means that cells can be isolated through a biopsy and then cultured in vitro. and then essentially produce the tissue of interest. In terms of cultivated meat, is the same process. However, it does require production of very large numbers, which means that those cultures will have to be translated from lab scale, which are flasks, to more of industrial scale, which are bioreactors. And to achieve that mimicry of meat, you do need multiple cell types. So that means muscle, fat, maybe even connective cells like fibroblasts. So how can it be produced? Well, in the production of cultivated meat, there are several, I like to call them puzzle pieces, because they are all very, very important and they make a whole, the whole being the cultivated meat product at the end. So these puzzle pieces are the categories of items that go into the production where a lot of considerations have to be made. So first we have the cell lines, then the media formulation for both proliferation of the cells, but also differentiation of the cells to achieve the muscle and the fat cells that are components. Then you have the scaffolds, which are necessary to grow the cells on, so something like microcarriers, but also if the final product that is aimed, is targeted, is a structured product, like a steak, then a scaffold to give it that structure would be necessary. And then, of course, we need the bioreactors and the bioprocessing in order to achieve those large numbers, cell numbers. And I'm going to go through each of these categories of items and give you a bit more insight. To start with cell lines, as I said, the aim is to achieve those cell types that are component of meat, which are muscle and fat. However, muscle cells or finely differentiated cells do have a very limited potential for proliferation, for expansion. So that means that in order to achieve the cell numbers that are required, we do most likely need to use stem cells that have the ability to differentiate into muscle cells or into fat cells. And this graph is actually taken from the Good Food Institute, from one of their surveys that was done to both academics and industry, to find out what kind of cell types are they using in their processes. And quite interestingly, the most common cell type are myosatellites, which are essentially unipotential stem cells that have the ability to form muscle, but then also fibroblasts, which are those connective cells that I've mentioned. And then other cell types can be mesenchymal stem cells that have the ability to form the fat, or pluripotent stem cells like induced pluripotent or embryonic stem cells that have the ability to form both the muscle and the fat. And to give you a bit of an idea, because I have talked about this quite a bit, that we do need very large numbers of cells. In one of the papers that was published by Mark Post, it was estimated that 1 ton of cultured meat would contain 10 to the 14 cells. So this is, I don't know how it translates, but it's 1 billion of 1 billion cells. So this type of cell number can only be achieved in bioreactors and in very large bioreactors. And this is just an example of a production flow, to give you a bit of an idea of how intensive the manufacturing could be in order to achieve these type of numbers. So in terms of scale expectations, because this is really important as well, so you can understand what is realistic and what is not. For therapeutics like proteins or monoclonal antibodies. Generally, these can be produced in bioreactors of 10,000 liters. So very large bioreactors, sometimes a few stories high bioreactors. Viral vaccines at the moment can be produced in 2,000 liter scales. These are the kind of maximums that we're talking about. Human stem cells for therapeutic purposes can be produced realistically in scales of around 200 or several hundred liters. So thinking about the cultured meat, where I said that in order to achieve those type of numbers and produce a significant amount of meat to be able to cater for the meat demand, and particularly thinking that in the production, the cells that would be used would be stem cells. The technology needs to advance to a point where it can move from several hundreds of liters that has been used for human stem cells to 10,000 liters that are common or standard for production of proteins. So we're still quite a way off in being able to produce these products so that they can be available, which is why, in a way, it's understandable why these cultivated meat products that have been approved so far are only available in high-end Michelin star restaurants at the moment. And sometimes there are very long waiting lists of several weeks to be able to source it. So the other category that I want to talk about is media. Because the process involves a step of expansion of the stem cells, but also a step of differentiation of those stem cells to achieve the muscle and the fat cells, there will have to be different media formulations for the growth and the differentiation. And generally, all media will contain things like salts, amino acids, vitamins, but then also proteins, growth factors, carbohydrates, etc. But the idea is that the media will be able to support the cells in both their expansion and differentiation. And at the moment, the problem is that the media is quite expensive, which also translates in a higher price for these products. However, most of these components can be sourced as food ingredients, and once that would be achieved, the cost will come down significantly, making cultivated meat products affordable. So in terms of manufacturing, excuse me, this is very similar to the process flow that I've showed a bit earlier, where in order to achieve the final scale has to go through several stages. And these stages are known as the seed train. And as I said, there will be a step of proliferation, but then there will also have to be a step of differentiation to achieve, to fully mimic the structure of animal meat. And of course, the expansion of the cells will have to be done in bioreactors. And this slide is just to give you an idea of the different types of bioreactors that are available. And all of these come with advantages and disadvantages. The most common type of bioreactor that is also used for the 10,000 liter scales that I've mentioned is the stirred tank bioreactor. So that one is the most established, but that doesn't mean that for this particular application is the most useful or the most suitable. And then I think I'm coming to the end of this presentation, but one of the last things I want to talk about is what are the options for growing these cells in those bioreactors that I've mentioned. So all the stem cells, or most of the stem cells that would be appropriate for production of cultured meat, are what we know as adherent-dependent cells. So that means that when we grow them in vitro, they require an attachment substrate or something to attach to that will help them survive and carry on all their biological functions. And that's something to attach to can be in different shapes. So they can be microcarriers, which are essentially, think of them like grains of sand, so micrometer-sized particles that the cells can attach and grow in when they are put in a suspension environment, like in a bioreactor. Some specific cell types, like pluripotent stem cells, can also grow without this attachment substrate as aggregates, or also known as spheroids. So in that case, you don't need an attachment substrate, but the challenge there is to control the size of those aggregates. Some of these companies, cultivated meat companies, are working towards adapting the stem cells to a suspension growth, which is a technique that has been achieved successfully in biotechnology in the past with CHO cells that are one of the mammalian cell lines that are used for production of proteins or monoclonal antibodies. And just to conclude, cellular agriculture is a very exciting and emerging area or discipline that has a lot of potential to produce animal source products that can be more sustainable. There are several concept products out there, and actually we're seeing a lot of developments in the cultivated meat space now with all the regulatory approvals. Yeah, hopefully in the next couple of years, we will see cultivated meat products on the shelves in different parts of the world. The technologies that are used to produce these type of products, some of them, I mean, they are imported from other disciplines like tissue engineering, so they're not new, but there are challenges that tissue engineering or therapeutics do not have, like the cost and availability, which are absolutely critical for cultivated meat products, for example, which means that there is a necessity for developing new technologies, a lot of potential for innovation in this space, and perhaps, you know, technologies that are imported from other disciplines like food processing could be very appropriate in this context. And I will leave it at that. Thank you so much for listening. Hopefully you found this very useful and insightful. And now I'm very happy to take any questions that you might have. As I said, feel free to reach out to me as well. And I can actually bring it back to that first slide so you can see my contact details there if you want to reach out. Thank you so much.
Great, thank you, Petra. So whilst I give Petra a chance to look through the questions, I see there are a few already. I just want to mention a couple of things that we've got coming up. Thank you for your kind words about the talk. It was very good. So for those of you who are interested in studying a degree with us here at Biochemical Engineering, we actually have an undergraduate virtual open day happening on the 23rd and then a repeat on the 24th of this month. I'll pop a link in the chat in a moment. You can sign up to that and join me and Dr. Jack Jeffries. If you'd like to join us in person, you can come along on the 28th or the 29th of June. It's an opportunity to come meet the team, have a look around the labs. And in fact, if you're considering doing an MSc, you can still come along to that because you'll get a chance to see our pilot plant. It's a unique facility in the UK, and it's a really great chance for us to show you around something which no other university has. So I'm going to pass you over to Petra. Petra, would you like to go through whichever questions in whichever order you would prefer to, and I'll just pop some links in the chat.
Yeah, of course. Okay, so I'll start in the order. So how about the cost when considering lab-grown food? So that is a really good question. The cost is very much linked to the production of these products. So whatever goes into the production will have an impact, which is why I said that, because most of these techniques have been imported from tissue engineering and biotherapeutics manufacturing, where basically cost is not necessarily a challenge, because you can't really put a cost on, on the health or life of someone. In this case, cost is really, really important. So media is generally the higher contributor to the cost. There are some initiatives in trying to replace all the components in the media with food grade components, which would bring the cost down significantly. Some groups, academic groups, and also companies are also working on trying to replace serum or some of the components in the serum, like growth factors or proteins, with food grade sources. So things like soy hydrolysates or other types of hydrolysates could have potential in replacing some of those, and this can bring the cost down even further. Other approaches to bringing the cost down can be through bioprocessing. So if the bioprocess or the manufacturing process is designed in such a way to increase the cell density, so obtain more cells per volume, then you can get away with a lower scale, so you can save on costs from that point of view. But then you can get more product essentially in a lower volume, so that can decrease those costs. Right, so moving on to the second question. Can mammalian cells be grown without a scaffold to produce a biomimic tissue similar to fungal mycelium? What is limiting innovation to understand why cell culturing is limited to unstructured cell biomass generation? Yeah, so that is a really good question. Different companies have taken different approaches. So for example, Aleph Farms that I've mentioned from Israel, they are doing a structured product, which is a bit more difficult to achieve because you still need to produce the cells in the large quantities in the first place. And then you have an additional step where you essentially need to achieve that structure. So their approach is to 3D print it. They still need to use, so they don't necessarily use a scaffold, but they still need to use a biomaterial, a bio ink, to mix the cells, and that bio ink helps achieve that type of structure. I guess a lot of the companies do end up going towards the unstructured or minced meat-like products because you don't have that additional step. So you only have to produce the large cell quantities of muscle and fat and then mix them in a certain proportion. So in some respect, it's a bit more simplistic. I mean, don't get me wrong, the whole process is still very complex, but you don't need that additional step of using another scaffold or using something else to achieve the structure. And in terms of innovation, there is a lot of potential for innovation. I did read about a company trying to use electrospinning to make sort of sheets of cells and then stack those to achieve that sort of 3D structure. So there are various approaches that can be used. The next question. That is a good one. Are all types of cells for cultivated meat cultured together? How are different types of cells structured together to get the final product? Yeah, that is a very good question. Unfortunately, not. I mean, if you would be able to produce both the muscle and the fat in the same bioreactor, so in the same pot, that would be ideal. That would be a very, a simplified process. But unfortunately, not, because because muscle has requirements, both in terms of the nutritional and in terms of the processing, which are quite different to fat. And also the lengths of time for that differentiation step are different. So for differentiating towards muscle, it takes around 7 days. For differentiating towards fat, to achieve mature fat, it takes 21 days. So unfortunately, for cultivated meat, if you do want to have both the muscle and the fat in there to achieve the mimicry, different processes are needed. One process to achieve each cell type. And then, I guess the second part of the question I've already answered. How are they structured together to get the final product? So if it is a unstructured product, you just mix the muscle and the fat in different proportions. So probably something like 80% or 90% muscle, and the rest would be fat or other cell types. And then for the structured, you would have to do a co-culture. So essentially seed muscle cells and fat cells in a similar proportion and allow them to essentially start combining to produce that final product, final structured steak, or what you're aiming for. The next question: what are the?
Sorry, just want to interrupt a moment, Petra. Just to let you know, we've got 3 minutes left. So perhaps if we focus on maybe 2 or 3 questions, so we can squeeze them for the end of the session.
Okay. What are the practical implications of using microcarriers in terms of safety? So this is a really good question. It depends what type of microcarrier you would use. So commercially available ones, which are generally made of polystyrene, so plastics, are not fit for purpose. There are companies working on producing edible microcarriers, food grade microcarriers, which would not have that risk. What could be the potential challenges facing lab-grown food industry? Well, the challenges are the cost and availability. So the cost and essentially the scale up of these processes, which is pretty complex. If those are achieved, then essentially we would be seeing those products on the market, and they would be affordable. Interesting. So why did you choose to focus on cellular agriculture, and what part of your job do you find the most interesting? That is a really good question. Thank you for asking that. To me, it's quite personal. So I focus specifically on cultured meat. And the reason for that is because I absolutely love animals. I mean, I have 2 cats of my own. I love them to death. They're like my babies. I would do anything for them. And I just can't stand to see animals suffering. So to me, the motivation was the animal welfare. Of course, I'm also, I'm also driven by the climate change, because I'm seeing what is happening and it is real. But I think to me, the main reason is animal welfare. What do I find most interesting as part of my job? Research. I mean, I love research, and the research I do, especially in this area, is because it's so new, it's very, very exciting. So anything that we do, anything that we discover is it is very, very exciting. And it's something that will it works towards making something better, having an impact, a real impact. And then I think, do we still have time for one last question?
I think we can squeeze in one more question, yes.
Okay. Would replacing farmed meat with cultured meat globally be an overall positive impact on the environment, considering the energy and water required? I think that is a fantastic question. I don't think we will be able to replace animal meat or animal farming in its entirety globally, maybe in 100 years, when the technology is good enough or sufficient. Maybe not even then. I think animals will still be around, but this industry, the cultured meat, is about offering options to consumers like me. I would eat meat, but because of that animal welfare element, I do not normally. I don't do it. I just stopped because of that. I have been to slaughterhouses, and I have seen how it looks like, and it has been horrific. So it's an personal choice for me not to. And I'm sure there are many other people like me out there which would still consume meat, but they have their reasons not to. So animals will still be around. I think the cultured meat is about taking some pressure off the animal farming so that we don't have those intensive animal farms, which are the ones that are creating all these detrimental effects. Right. I kind of feel bad because there are a few other questions in there. Potentially.
And there are some excellent questions as well. So
Yeah.
there's some really good. But unfortunately, we have run out of time. But I'd like to thank everyone who asked those questions. Those were some of the best questions we had so far. So it's really great to hear them come through. Thank you for your time. Thank you for joining us. If you're able to come and join us in person at one of our sessions, it'd be great to welcome you at UCL. You can see the laboratories, and of course our two different types of laboratory. We do have mammalian labs as well. The links are in the chat. What I will do is I'll send a link out to people as well by email so you can come along to that. But in the meantime, thank you very much for joining us. I hope you have a great weekend. Petra, thank you very much for your time in giving this talk. It's been great. And have a fantastic day. Bye-bye, everybody.
Thank you so much, everyone. Take care.